Hey everyone, it is January 15th, 2019. My name is Desiree Warner, and I am currently working on my assignment, the Bacterial Enumeration, Dilutions, and plate counts. And I've started my experiment. As you can see, I'm wearing my protective gear, and this is my workstation that I have set up here. I believe you can see everything I have. I have my test tubes, my pipette, my diluted water, my alcohol, my agar solutions, and just putting on my gloves. And my petri dishes, again my test tubes sitting in the test tube rack. So what I'm gonna do, what I've done so far is, in the interest of time, I figured I will do the experiment and then do the recording, because we have a 4 to 5 minute limitation, I believe, or 6 minutes limitation for the video presentation. But I'm gonna go through what I did. So I mixed the amount of yeast in the warm water that they said it was a quarter teaspoonful in the warm water, and two tablespoonfuls of water, and then mixed it. I let it sit for 10 minutes, and here it is. This is the yeast that I made. And see, the bubbles there, it's frothing. The bubbles there, as you can see. Then in each test tube, this is test tube 1, I put in distilled water as instructed of 2.25 milliliters. And I label these, each test tube 1, 2, 3, test tube 4, 5, 6, that's 10-1, 10-2, 10-3, 10-4, 10-5, 10-6. And so each test tube got 2.25 milliliters. And then the yeast solution, I pipetted from the yeast solution, and making sure to clean the pipette between each pipette and between each procedure. I pipetted 0.25 milliliters of the yeast into 10-1. Then from 10, I cleaned the pipette. Then from 10-1, I pipetted 2.5 to 10-2. Then I cleaned the pipette. Then I pipetted 2.5 again from 10-2 to 10-3, transferring. Cleaned the pipette. Pipetted 0.25 milliliters from 3 to 4. Clean. Transfer from 4 to 5. Clean. Transferred again 2.5 that solution from 5 to 6. So in other words, what they're trying to get us to do is to work with a gradation of concentration of the yeast solution. So from 6 would be highly concentrated. From 10-1 would be highly concentrated, 10-2 less, 10-3. So the higher the number we get, the less of the concentration. Then prior to the pouring, I prepared my agar solution, which would have been in these test tubes. I labeled them, as you can see. I also labeled the covers, the respectful numbers, 10-1, 10-2. And then I poured the agar solution, poured into each petri dish. And then I poured, I dropped 4 drops of 10-1 respectively into 10-1 petri dish of the agar solution, 10-2 into the 10-2 petri dish, 10-3 into 10-3 petri dish, the 10-4 test tube solution into the 10-4 petri dish, 10-5 into the 10-5 petri dish, and of course the 10-6 would go into the 10-6 petri dish. And we are instructed to now put it into incubate wait for 12 hours, and then flip the petri dish to, I guess, get the cultures to fully grow and expand. And after that, after the 48-hour wait, we are to start counting and calculating. I hope I did not miss anything. The calculations will be done, and it says here in my experiment, wait 12 hours, then invert the dishes to incubate for an additional 36 hours. Like I said, it's 48 hours in addition. And then I am going to take pictures of the solution, count the colonies, and present this in my lab. And that's it. Thank you, everyone. Again, my name is Desiree Warner. It is January 15th, 2019.